Hi, my name is Bhoomika, and in this chapter we're going to study about the basic physiology of digestion. I have already done a chapter in which I've given note of all the background data required for this, so you can check that out. I think we should start. We're going to begin with the digestion of carbohydrates, and obviously this whole presentation will be referring to all the biomolecules separately, so I'm going to start with carbohydrates first. The mechanical and chemical digestion of carbohydrates, it begins in the mouth. Chewing, which is also known as mastication, it crumbles the carbohydrate foods into smaller and smaller pieces. The salivary glands in the oral cavity secrete saliva that coats the food particles. Saliva contains the enzyme salivary amylase. This enzyme breaks the bonds between the monomeric sugar units of disaccharides. So basically the bonds that break are alpha 1,4 glycosidic bonds, oligosaccharides, and starches. The salivary amylase breaks down the amylase and amylopectin into smaller chains of glucose, which is known as dextrins and maltose. Now, the increased concentration of maltose in the mouth that results from the mechanical and chemical breakdown of starches in whole grains is what enhances the sweetness. Only about 5% of the starches are broken down in the mouth. This is a good thing, as more glucose in the mouth would lead to more tooth decay. Now, when the carbohydrates they reach the stomach, no further chemical breakdown occurs because the amylase enzyme it does not function in the acidic conditions of the stomach. It has got a very high pH. It is near 1, obviously because of the presence of the HCl. So here it inactivates. But mechanical breakdown is ongoing. The strong peristaltic contractions of the stomach, they mix the carbohydrates into more uniform mixture of chyme. Now, whenever a food bolus it is pushed down the esophagus, it remains a bolus until the stomach, and after passing through the stomach, it is known as chyme. The chyme is gradually expelled into the upper part of the small intestine. Upon the entry of the chyme into the small intestines, the pancreas they release the pancreatic juice through a duct. This pancreatic juice it contains the enzyme pancreatic amylase, which starts again the breakdown of the dextrins into shorter and shorter carbohydrate chains. Additionally, enzymes are secreted by the intestinal cells also, which lie in the villi. These enzymes, known collectively as disaccharides, are sucrase, maltase, and lactase. Sucrase breaks sucrose into glucose and fructose molecules. Maltase breaks the bond between the two glucose units of maltose, and lactase breaks the bond between the galactose and glucose. Once carbohydrates are chemically broken down into single sugar units, then they are transported into the inside of the intestinal cells. So, whenever this question is asked in the examination, it may either be asked as state the complete process of digestion, in which you will be writing about all the three biomolecules. But if it is asked specifically that discuss the digestion of carbohydrates, then of course you're going to mention it that this much of digestion takes place in the mouth, this much of digestion takes place in the small intestines, and in the under the action of pancreatic juice as well. So make sure you write down all the conversions into equation form. As you can see here, as it is written, that maltase breaks the bond between the two glucose units of maltose. So you're supposed to write maltose in presence of maltase becomes glucose, and glucose. So you literally give the examiner a review that you have your concepts cleared this way, and you get more marks. So let's move on. What we've got is digestion of proteins next. The protein digestion begins in the stomach by the gastric juice. So there's no digestion of protein that takes place in the mouth. Role of the gastric HCl, it causes denaturation of proteins. Denaturation of proteins means breakdown of the chains. It activates the pepsinogen to pepsin. Pepsinogen is the inactive form of pepsin, so HCl helps in activating it. It makes pH in the stomach suitable for the action of pepsin. Now let's talk about pepsin. It is an endopeptidase. Endopeptidase is an enzyme which works on the peptide bonds which is present on the inner side, acting on the central peptide bonds in which the amino group belong to the aromatic amino acid, that is phenylalanine, alanine, tyrosine, and tryptophan. It is secreted in an inactive form called pepsinogen, and the optimum pH required is 1.5 to 2.2. It is activated by HCl and then by autoactivation. We are also going to talk about an enzyme called renin, which is a milk clotting enzyme, and it releases a single amino acid. It is present in the stomach of infants and young animals, and the optimum pH is 4. It acts on casein, converting it to soluble paracasein, which when binds with calcium ions forms the insoluble calcium paracaseinate. And then calcium paracaseinate is then digested by pepsin. You must have noticed that small infants, when they eat something, they're supposed to be patted on their backs, and then some white colored curdling mixture comes out of their mouth. That is calcium paracaseinate actually. Gelatinase, it is an enzyme that liquefies gelatin. The end products of protein digestion in the stomach are proteoses, peptones, and large polypeptides. Now, the next digestion will obviously take place in the small intestines. Digestion of proteins is completed in small intestines by proteolytic enzymes present in the pancreatic and intestinal juices. So first we'll talk about the digestion that occurs through the pancreatic juice. So the first enzyme that works upon the polypeptides that have come from the stomach is trypsin. It is an endopeptidase that hydrolyzes central peptide bonds in which the carboxyl group. It belongs to the basic amino acids. For instance, arginine, lysine, and histidine. It is secreted in an inactive form, of course, the trypsinogen, whose optimum pH is 8. It is activated by enterokinase enzyme. Enterokinase is actually secreted by the small intestines. The second enzyme from the pancreatic juice is the chymotrypsin or the chymotrypsin. It is an endopeptidase that hydrolyzes central peptide bonds in which the carboxyl group belongs to an aromatic amino acid. It is secreted in an inactive form called chymotrypsinogen. It is activated by trypsin. So trypsin is actually activated by enterokinase, and then trypsin in turn activates chymotrypsinogen. Its optimum pH is 8. We also have another enzyme called the elastase. It is an endopeptidase acting on the peptide bonds formed by glycine, alanine, and serine. It is secreted in an inactive form called proelastase. It is activated by trypsin and it digests the elastin and collagen. Optimum pH is 8. And fourth and the final enzyme that is found in the pancreatic juice is carboxypeptidase. It is an exopeptidase. That means it works on the bonds which are present on the outer ends. It hydrolyzes the terminal or the peripheral peptide bonds at the carboxyl terminus at the end of the polypeptide chain. You can remember this as here you've learned carboxypeptidase. So that means it will hydrolyze peptide bonds at the carboxyl terminus. It is secreted in an inactive form called procarboxypeptidase, and again it is activated by trypsin, and the optimum pH is 7.4. Let's talk about intestinal juice. Firstly, aminopeptidase, which is an exopeptidase that acts on the terminal peptide bonds at the amino terminus of the polypeptide chain is there. Then we've got the tripeptidase, which acts on the tripeptides. It releases a single amino acid and a dipeptide. Then to digest these dipeptides, we've got the dipeptidase. It acts on the dipeptides and it releases 2 amino acids. The end products of the protein digestion in the small intestines are amino acids, of course. Okay, so before beginning the next topic, I would like to tell you that you just need to know the general pH of each of the following organs. You don't need to learn it for separate enzymes. Like here it's given carboxypeptidase's pH is 7.4, and then in the previous slides also it was given for each enzyme. You don't need to remember all of that for your school examinations, but again it may be asked in the competitive examination. So here also I would recommend all of you to write down each and every conversion into an equation form after you've written it in theory. It just makes everything seem clear and the examiner is convinced that you know what's going on. Okay, so now let's move on to the digestion of fats. In the stomach, when you swallow, the food pushes down on your esophagus. There are pipeline going through your throat and neck, and then it falls into a bag-like organ, which is the stomach, where the pepsin enzyme further breaks down the food proteins. We have studied all of this. Salivary lipase is also still active at this point. It continuously secretes through saliva and naturally accumulates in the stomach, where it hydrolyzes fats. So this is a kind of an enzyme which works onto the fats, and it hydrolyzes fats. When the lipase and pepsin have done their work, the resulting mush goes to the pyloric valve. We had done the position of the pyloric valve in the digestive system, and separating the stomach from the small intestine. Now in the small intestines, your liver and the pancreas, they fill the small intestines with the substances that will do serious chemistry with fats and carbohydrates. The bile salts from the liver, they liquefy the fats in the mush. Meanwhile, the juice which is secreted by the pancreas carries pancreatic lipase, which breaks down the fat molecules down to the simplest forms. Also, some intestinal lipases also work upon this, and finally it is absorbed in the form of chylomicrons. We'll do the absorption process in the next lesson, of course. So that was about the basic physiology of digestion. If you want to understand this in Hindi, then I'm also going to update this in the other course that will follow this particular course. It'll just increase your understanding if in case you're comfortable with the language. I'm not, of course, pushing you good too. Again, I'm saying, if this is asked in the examination excuse me, if this is asked in the examination, you are supposed to write down everything in theoretical form and then mention everything in the equation form. Write down the enzymes, write down the pH, everything. If you miss even a slight thing, it manages upon your marks. And especially in boards, in the competitive examinations, you've got MCQs, so it may get confusing, but if you learn it with a tactical approach, you'll be able to do it. So in the next lesson, we'll be discussing about absorption processes. If you like this tutorial, then you may follow my other tutorials as well. You can rate me, review me, and recommend this lesson. Thank you.